So thank you so much. So hi, everybody. My PhD work explores circadian disruption. I'm just going to actually get my laser pointer. So I explore circadian disruption in prenatal immune activation in mice, and I'll explain the implications this has for neurodevelopmental disorders, including schizophrenia and autism spectrum disorder, or ASD. So many individuals with schizophrenia and ASD exhibit severe sleep and circadian disruptions. So actograms here, which are visual representations of activity over 24 hours. We can see here that the actograms for the patients with schizophrenia show clear signs of circadian misalignment, ranging from phase advance and delay patterns, while others show highly fragmented and irregular activity patterns compared to the matched controls. And this paper I cited below describes the disrupted activity patterns in ASD. In addition, many parameters associated with sleep are also significantly disrupted, denoted by the red stars, in individuals with schizophrenia on the left and in individuals with autism on the right when they're compared to healthy matched controls. And this made us wonder, what is the relation between circadian disruption and neurodevelopmental disorders? Well, we know that exposure to maternal infection has been associated with a variety of neurodevelopmental disorders. Exposure to the flu in the first trimester increases the offspring's risk for developing schizophrenia by up to 7-fold. And a viral infection, also in the first trimester, increases the offspring's risk of developing ASD by almost 2-fold. And we can actually model prenatal immune activation in a mouse by exposing pregnant dams to a synthetic viral mimic called poly-IC at embryonic day 9.5, which is attributed to early gestation in humans. And we studied the circadian rhythms in the resulting offspring. So briefly, what is poly-IC? Poly-IC is a synthetic analog of double-stranded RNA, and it is recognized by the transmembrane protein toll-like receptor 3. And poly-IC mimics a viral infection, and it triggers the rapid production and release of many pro and anti-inflammatory cytokines and chemokines in the maternal environment at the maternal-fetal interface. And these cytokines and chemokines invade the fetal body and the fetal brain and affect fetal development. So we first wondered if we could recapitulate these circadian disruptions that are originally observed in patients in our poly-IC exposed offspring. So we first induced maternal immune activation in pregnant dams. Then we gave adult offspring access to a running wheel, and we analyzed wheel running parameters under standard lighting, constant dark, and constant light. So again, I'm showing you actograms, which are activity over 24 hours. The saline exposed mice are on the top, the poly-IC exposed mice are on the bottom, and here you can see the exposure to the 3 different lighting conditions. And we found various differences between the poly-IC and saline exposed offspring, but I'll only show you the most striking difference that we saw, which was that prenatal immune activation induced an increase in daytime activity across all 3 lighting conditions. So what does this mean? So under standard lighting, the poly-IC exposed mice were more active in the day than the saline exposed mice. And under the constant conditions, the poly-IC exposed mice had more subjective day activity than the saline, and similarly also in constant light. And the female offspring had a much milder phenotype than the males. So for our next experiment, we explored the effects of environmental circadian disruption. So we probably all know that exposure to artificial light, especially at night, and the ability to quickly travel across time zones certainly has a negative impact on our circadian clock, which has evolved to the natural rhythms of the light-dark cycle of day and night. So also one hypothesis for the development of neurodevelopmental disorders is that there are a number of accumulating risk factors, and these will predispose an individual for different kinds of neurodevelopmental disorders. And we hypothesized that environmental circadian disruption could be a risk factor that one is exposed to in their life that could affect the course of the disorder. And we wanted to explore if environmental circadian disruption interacted with prenatal immune activation in mice. So we first explored this interaction on a behavioral level. So we induced maternal immune activation in pregnant dams. Then 3-week-old offspring were subjected to 4 weeks of either standard lighting or constant light LL, which is our environmental circadian disruption. And then we performed a number of behavioral tests. So first we used the Barnes maze to assess spatial learning and memory. So rodents have a natural aversion for elevated and open spaces, bright lights, loud sounds. So this motivates them to seek shelter in the escape box, which is placed under one of the 20 holes on the apparatus. And mice were trained to locate this escape box using a configuration of visual cues located around the testing area. And on the test day, this box is removed and the time spent in the target location is assessed. So on the test day, the poly-IC exposed mice spent significantly more time in the target area than the saline exposed mice. Also, the poly-IC LL group performed significantly more entries into the target area than the saline LD group. In line with these results, the poly-IC LL group made fewer errors on the test day, and they had a shorter latency to reach the target area. Now, the Barnes maze was designed to measure spatial learning and memory, but perhaps the outcome we see here is rather a result of increased impulsivity in the poly-IC exposed mice. The elevated plus maze was used to assess anxiety-like behavior. And here, if we look at the data, we see that LL caused in the saline exposed mice to exhibit less anxiety-like behavior. And as seen here in the percent time spent in the open arms and in the percent entries into the open arms. But surprisingly, constant light LL did not have this effect in the poly-IC groups. Additionally, females did not show these differences. We also performed a social interaction test, and I'll only go over the social preference phase of this test, where the test mouse can explore either another mouse or an object. And mice typically prefer to spend more time around another mouse versus an object. So we can see here that under standard lighting, the poly-IC exposure led to a decrease in social preference, which is consistent with the literature. But this group difference was not seen between the LL groups, so saline LL and poly-IC LL. So clearly LL exposure seems to be affecting saline and poly-IC mice differently. And these effects were not observed in females, which I showed here. So clearly on a behavioral level, LL exposure differentially affects saline and poly-IC exposed mice, which made us wonder, what is the cellular mechanism that underlies these effects? So we decided next to study microglia. So not only is embryonic day 9.5 when we inject poly-IC into the pregnant dams, but it's also when microglia colonize the fetal brain. And embryonic microglia then expand and colonize the central nervous system until they acquire their mature characteristics in adulthood. And these microglia remain in the brain until adulthood, highlighting the importance of embryonic microglial development. And for us, microglia are also an interesting topic to study because microglia have circadian clocks. So we use the same experimental design as before, but we ended up staining the brains for microglia using IBA1. And these are the results in males. So Overall, poly-IC exposure led to decreased arborization compared to the saline mice, which is indicative of a more reactive profile. And when looking at morphology, the poly-IC groups showed a trending effect for a more reactive microglial profile compared to the saline. But in both groups, interestingly, the LL exposure surprisingly led to a less reactive microglial profile. And I won't show you the data from the females today, but they had a different profile than the males. So in males, the poly-IC led to a more reactive microglial profile, whereas constant light exposure surprisingly led to a less reactive microglial profile. So our follow-up question to this was, what is the molecular mechanism that underlies these effects? So we have the same experimental design as before, but this time we microdissected out the hippocampus from poly-IC and saline exposed male and female after the standard light or the disrupted light. And our goal was to analyze differences in the hippocampal transcriptome. And I'm currently in the process of analyzing these data, but I can quickly show you this Venn diagram of differentially expressed genes between groups. So as we can see here in yellow, under standard lighting, exposure to poly-IC led to around 2,000 genes, protein coding genes, to be differentially expressed. In the poly-IC exposed mice, exposure to constant light led to around 500 genes to be differentially expressed, whereas constant light did not have this effect in the saline mice. So much more analysis to come on this front. But overall, we hope to identify the mechanisms that underlie this interaction between circadian disruption and neurodevelopmental disorders, which could have the implication in informing the development of circadian-based therapies. And with that, I'd like to thank my supervisors, Nico Sermakian and Alit Srivastava, and their respective labs, and also a number of helpful individuals at the Douglas Mental Health Institute and McGill University. Thank you.
Thank you, Tara. Very interesting presentation. Hopefully they are not only helpful and they are healthy also, your colleagues. So, questions for Tara. So while people are thinking, let me ask one about your model. I'm just wondering, I'm not familiar as well with poly-IC model. How does it compare with LPS model, and also how with your dosage, or in your case, how sick the mothers were after you injected them?
Yes, so just going back to my, yes, exactly. So there are many different models to study maternal immune activation. And we decided to go with this model because poly-IC mimics a viral-like infection, whereas LPS is more bacterial. And just because of the association between schizophrenia and autism with viral infections, we decided to go with poly-IC. And your other question was about dosage. We actually had a pretty low dose of poly-IC. So it was 5 milligrams per kilogram. In the literature, people use as low as 5 and as high as 20. And this is a single injection, which is quite remarkable that a single injection during can affect the pups for their lifetime. And you had another question as well about...
I mean, what do you see anything clinically, if you like?
Oh, right, yes. So, yeah, so it induces sickness-like behavior in the moms over 24 to 48 hours. So, for example, reduced locomotion. Some studies have reported decreased feeding within that range. And of course, sometimes there is a fever and there's this rapid release of cytokines and chemokines. And we actually have a validation study where we collected maternal blood and looked at the cytokines and chemokines, and we found an increase in pro-inflammatory and some anti-inflammatory cytokines in the maternal blood after our poly-IC injection.
Okay, thank you. Any questions?
I think Dr. Chakrabarty has one.
Okay, I was wondering, I was worried we were a bit over time, but we have.
Oh, sorry.
No, we're good. We're good. We have a few more minutes. Okay. Great talk. It's nice to really, every time I see you present, there's just a significant chunk more data. It's really fun. So I don't want to put you too much on the spot, but I'm very intrigued by the sex differences that you didn't present. And could you give us a little glimpse into what it might look like? You don't have to give me the whole story if you want to, but just a little bit.
With regard, well, just overall.
Like, overall, or the microglia, for example, I think.
Oh, okay, yes. So I don't know if I actually have a slide on this. Oh, no, I don't. But in the microglia, basically, we saw a different profile. We found that there were differences in density, microglial density, between the saline LD group and the poly-IC LL group. So those two hits, the poly-IC and the LL exposure, led to differences in microglial density. There were also some differences in the shape of the microglia that did not result in overall differences in morphology. There's definitely something going on with the sex differences that we hope to uncover in the transcriptomic study.